My name is Jishun Pallaru, and my research question was, does HYPE-mediated AMPylation affect BiP's translocation to the cell surface? My research goal for this project was to examine a specific protein-protein interaction in depth and attempt to characterize it better. Now, to understand this project, we must first examine the proteins involved and how they interact. HYPE, or Huntington yeast interacting partner E, is a human ampylator. What does that mean? It means that it covalently attaches an adenosine monophosphate, or AMP, to its substrates. One such substrate is BIP, the master chaperone of the unfolded protein response, or UPR. The UPR can induce apoptosis in cells undergoing severe ER stress, so cancers tend to suppress this apoptotic branch of the UPR in order to avoid cell death. During ER stress, BIP is also translocated to the cell surface. If HYPE-mediated ampylation affects the translocation of BIP to the plasma membrane, this would suggest a direct role of HYPE in cancer cells and a new line of therapeutics to help treat cancers. I hypothesize that this ampylation does, in fact, have an effect on the translocation of BIP. Now, wild-type HYPE is intrinsically inhibited by the glutamate at its 234th residue. As a result, in order to induce overampylation to test my hypothesis, I needed to use a mutant. If the glutamate is mutated to a smaller residue, ATP can then fit into the binding pocket, which can then be covalently attached to a substrate. So, I used a mutant that has the glutamate changed to a glycine, referred to as E234G, or just EG-HYPE. I also, as a control, used a mutant that is completely inactive due to the mutation of a histidine residue that is essential to ATP catalysis, referred to as E234G H363A HYPE, or EGHA HYPE. All mutants were then tagged with GFP in order to verify transfections. The first step of my procedure was to transform plasmid DNA into competent DH5 alpha E. coli. DH5 alphas are excellent at producing more plasmid, so that I could take the little amount of plasmid that I used to transform and produce enough to transfect into human cells. Simultaneously to this transformation, I cultured HeLa cells in a 75 centimeter squared flask so that they would be confluent in roughly 2 days. Once the cells became fully confluent, I split them into 4 6-well plates so that after they grew overnight, they could then be transfected with the purified plasmid DNA and left to express for about a day. Following the overnight expression, the cells needed to be stressed. I chose thapsigargan as my stressor. Following an overnight stress with thapsigargan, the cells were washed and then fixed with paraformaldehyde. Now, the main purpose of this experiment is to differentiate between intracellular and membrane proteins. We can test for this by permeabilizing our cells. Any permeabilized samples, when stained with antibody, will fluoresce depending on the amount of protein inside the cell. However, in unpermeabilized samples, the antibody cannot pass through the membrane and can thus only recognize surface proteins. Finally, after this antibody treatment, the cells were left to dry overnight before being imaged by a fluorescent microscope at the appropriate wavelength. These are rough sketches of the expected results. I expected that unstressed samples would not have any BIP translocated to the cell surface, and thus BIP could only be found in those permeabilized samples. However, in stressed samples, once BIP is translocated to the cell surface, it should be detectable in the unpermeabilized sample, as it is now located at the cell membrane. When comparing the different constructs of HYPE, I expected that in the unpermeabilized stress samples, EGHA HYPE will have some BIP at the cell membrane, but not very much. But in the wild-type and EG HYPE samples, there will be much more BIP at the cell membrane, translating to a much stronger fluorescent signal. Here we have side-by-side comparisons of the unstressed and stressed cells with both DIC and GFP images. Looking at the DIC images, we observed a greater number of rounded cells in the stress sample, suggesting that our thapsigargan treatment was successful, because rounding is a phenotype of stress. When looking at the GFP images, the empty GFP and EGHA transfections were both very successful, but the wild-type and EG HYPE are expressed at a much lower level, likely due to a much less clean purification. When looking at the fluorescent microscopy, almost no fluorescence appears in the unstressed and unpermeabilized samples, which is to be expected, as BiPs should not be translocated to the cell surface under non-stress conditions. In all of the stress samples, however, there is at least some fluorescence, indicating that BiP is indeed translocated to the membrane under stress conditions. When comparing EG HYPE and wild-type HYPE to the controls, despite low levels of expression, there appears to be a greater amount of fluorescence, suggesting that overampylation possibly increases BiP translocation to the membrane, which would then support my hypothesis. In the second pass of this experiment, I will repurify the plasmids that expressed poorly and verify the purity of these concentrations using a 1% agarose gel. Next time, I would also plate onto cover slips when I do my 6-well plating, so that clearer, more focused images can be taken of my cells instead of the diffuse imagery shown here. Future experiments include treating BiP with an anti-AMP antibody in order to verify BiP's ampylation status at the membrane, and examine BiP translocation in non-cancerous cells to see how it differs, if at all, from translocation in cancer cells. Thank you.